Thyroid gland. Thyroid gland is one of the largest endocrine glands in our body. It is present below the larynx on each side of the trachea. It is about 15 to 20 grams in weight in adults and secretes two major hormones, thyroxine, that is T4, and triiodothyronine, that is T3. Function of these hormones is to increase the metabolic rate in our body. If these are absent, then the basal metabolic rate decreases 40 to 50%. And if they are present in excess, then the BMR increases 60 to 100% than the normal range. Secretion from the thyroid gland is mainly controlled by thyroid stimulating hormone, which is secreted from the anterior pituitary gland. Thyroid gland is made up of many closed follicles. These follicles are filled with colloid and are lined by cuboidal epithelial cells. Major constituent of this colloid is a large glycoprotein called thyroglobulin. Thyroid hormones are synthesized within the thyroglobulin molecule, and they remain part of it during the synthesis as well as storage in the follicular colloid. Now let's see the formation of these thyroid hormones. Iodine is required for the formation of thyroid hormones. Iodine is taken as iodide and absorbed like chlorides in our body. Basal membrane of the cells of thyroid gland has iodide pumps which can concentrate iodide from the blood into the cells by means of active transport. This is known as iodide trapping. Rate of trapping is influenced by many factors, most importantly the concentration of TSH. Thyroglobulin molecule is synthesized within the cells of thyroid gland. Each molecule of thyroglobulin has 70 tyrosine amino acids. Iodine binds with these tyrosine amino acids to form thyroid hormones. First step in the formation of thyroid hormones is the conversion of iodide into an oxidized form of iodine, only which can combine directly with the tyrosine amino acid. Oxidation of iodide is carried out by enzyme peroxidase and accompanying hydrogen peroxide located in the apical membrane of the cell of the thyroid gland. Now next step is the organification of thyroglobulin molecule, that is binding of iodine with the tyrosine amino acid, and this is carried out by the enzyme iodinase. Tyrosine is first iodized into monoiodotyrosine and then into diiodotyrosine. Then these iodotyrosine residues become coupled with one another to form T4 and T3. All these remain part of the thyroglobulin molecule, that is MIT, DIT, T4 and T3. And finally, a thyroglobulin molecule contains up to 30 T4 molecules, that is thyroxine molecules, and a few triiodothyronine molecules. T4 and T3 are cleaved from the thyroglobulin molecule, and these free hormones are released into the blood. But for this, thyroglobulin is first enclosed into pinocytic vesicles within the cells through pseudopodial extensions of the apical membrane. Lysosomes then fuse with these vesicles. Multiple proteases present in the lysosomes digest the thyroglobulin molecule and release T4 and T3. T4 and T3 then diffuse through the base of the thyroid cell into the surrounding capillaries. Monoiodotyrosine and diiodotyrosine are also freed from the thyroglobulin molecule, but they are not secreted into the blood. Iodine present in these molecules are cleaved by the enzyme deiodinase in order to recycle iodine. Thus, this step is a very important step. Otherwise, person becomes iodine deficient. From the blood, T4 and T3 are transported to the tissues in combination with plasma proteins, mainly globulin. These plasma proteins have high affinity for these thyroid hormones, and among these hormones, they have more affinity for T4 than T3. Therefore, T4 is released more slowly than the T3. In the tissue cells, they bind with intracellular proteins and are stored and used slowly. T4 again binds more strongly with these intracellular proteins than the T3. Therefore, on reaching tissues, almost all T4 ultimately gets converted into T3. Thus, T4 is mainly present in blood. Almost 93% of the thyroid hormones in blood is T4, but in the tissues, T3 is mainly present. Functionally, both T3 and T4 are the same. They only differ in the rapidity and intensity of their action. T3 is 4 times more potent than T4. Thyroid hormones have a slow onset but long duration of action. There is a latent period before these hormones begin their activity. This latency and prolonged period of action is due to their binding with the proteins in plasma and tissues, followed by their slow release from these proteins. This is also due to the manner these hormones perform their function within the cells.